Hi, my name is Tyler Hamer, and for the last 3.5 years under Dr. David L. Trumper, I've been working on an agile, low power attitude control actuator, or alpaca. Besides referring to the animal, alpaca refers to the device shown here, which is a spherical dipole permanent magnet, or a magnet with a north and a south pole. We place such a magnet inside a satellite. If we go to rotate the magnet, we have to apply a torque, and equal and opposite torque is then applied to the satellite's body, which you can use to control the orientation of the satellite. So far, we've been able to demonstrate that we can control the sphere's position in 3 space. So here we're going to have the sphere track a sine wave in the x, y, and z directions. Additionally, we've been able to show while having the sphere levitate that we can spin the sphere about the x, y, and z axes. So here, the sphere is spinning about the x-axis, the y-axis, and finally the z-axis. So if this interests you, please stop by my poster. I'm happy to give you more information.
Hi, my name is Anthis, and I'm a second-year master's student in the Precision Motion Control Lab. In our lab, we use magnetic actuators extensively, and in order to drive them, we need transconductance amplifiers, which provide a current to our actuators as specified by a voltage input. I've designed a linear transconductance amplifier for general use in our lab by employing classical loop shaping techniques, as well as electrical and thermal analysis. I chose to implement a lead lag compensator for system control, whose crossover frequency and breakpoint frequencies can be easily adjusted for a particular actuator by choosing the values of 3 passive components. In my electrical design, I included many safety features to prevent actuator and amplifier damage, and also added a buffered feedback signal output so we can display that signal for troubleshooting. These amplifiers are a simple, low-cost method of powering magnetic actuators. They are widely configurable and robust, and will hopefully save us time and money as they are used to power various setups in the lab.
Hello everybody, my name is Joshua Sohn. I'm an undergraduate student working under Moses Na and Professor Hogan at the Newman Lab. In this project, we attempt to understand why and how humans are so much better at manipulating complex objects when compared to robots, despite our hardware being so much worse. We contend that this can be explained by the internal model hypothesis, which says that prediction based on some sort of internal model plays a prominent role in how our motor control works. To do this, we analyzed how humans operate an object that is extremely complex, yet still very common, the whip. Since everything was remote, we simulated how a human arm cracks a whip by creating a model with a Python simulation package named Mccue. Because the whip is such a complex object, it's almost impossible to simulate an exact replica of the whip as one continuous object. So we simplified it into point masses connected by massless cylinders. The goal of my UROP was to explore whether mass tapering makes any quantitative or qualitative difference in the movement of the whip, specifically the maximum velocity of the tip if and when it cracks. This research is fundamental to understanding the mechanism behind human movement, and we hope it can be applied to improve robots in the near future.
Hi, my name is Caroline Mccue, and my poster is on particle-assisted protein crystal nucleation towards crystallization in downstream protein purification. A major challenge in manufacturing protein-based drugs is the high cost of consumables used for chromatography. As a result, there has been considerable interest in using crystallization as a protein purification method instead. However, crystallization requires high concentration of proteins, and nucleation of protein crystals is an extremely slow process. This research aims to address these two challenges using functionalized nanoparticles. Here, you can see a time-lapse of proteins crystallizing within microdrops. On the left is the control case, protein solution with no nanoparticles added. On the right, the protein solution contains functionalized nanoparticles. Under the exact same conditions, you can see that the drops containing functionalized nanoparticles crystallized much faster. If you'd like to learn more about this study, please come check out my poster. Thank you.
Atomic force microscope is a nanoscale topography imaging tool for surface characterization. The development of an AFM as a mechatronic system requires trained engineers with multidisciplinary backgrounds. Traditional AFMs are too expensive for hands-on education. In this work, we developed a modular low-cost, less than $4,000 AFM for precision instrumentation education. The educational atomic force microscope design has 9 primary subsystems including a piezo buzzer-actuated scanner, an active cantilever probe, a Wheatstone bridge amplifier, a signal generator and demodulator circuit, a National Instrument MyRio, a stepper motor driver, a coarse positioner engagement system, a buzzer scanner driver circuit, and an optical microscope. An educational workshop was offered at MIT during 2020 IAP on the AFM topic with 40 registered participants. We plan to develop a full semester curriculum centered around this system.
Hello everyone. I'm going to take this minute to give you a short tour of what my work is about. We at the Laboratory for Bio-Inspired Photonic Engineering work on different classes of materials that all have interesting optical properties. Some of us have been focusing on tunable optical properties, for instance, color-changing materials. I mostly work on stimuli-responsive photonic hydrogels, and in my poster I'm going to show you how we can start from a nanoscale material to build a functional material that shows dynamic and responsive behavior on macro scale. And here is a sneak peek of those materials. I'm either stretching the material, compressing, heating, or squeezing. Thank you for watching this.
Hi, my name is Ryuichi Iwata. I'm a visiting scholar at Device Research Lab. I'm currently working on bubble dynamics in water splitting to improve hydrogen production efficiency. Bubble growth and departure are ubiquitous phenomena in gas-evolving reactions which govern the overall energy and mass transport. However, an in-depth understanding of the relationship between bubble dynamics and electrochemical processes, in particular the wettability effect on a gas-evolving porous electrode, remains elusive. In this work, we investigate the bubble dynamics and overpotential observed during alkaline water splitting on a PTFE deposited nickel porous electrode. The insight gained from this study not only shed light on the fundamentals among electrode wettability, bubble dynamics, and overpotential, but also provide design guidelines for porous electrodes to enable high-performance gas-evolving reactions.
Hi, my name is Ben Miller, and I'm a PhD candidate in the Laboratory for Biologically Inspired Photonic Engineering. My research is based around materials like this, which change color as you stretch them. These are inspired by nature, specifically a phenomenon called structural color, where color is produced not by absorption, but by nanoscale structures. I've developed a method to produce these materials that's low cost, scalable, and allows you to print images and patterns in full color. This technique is actually based on an early type of color photography that won a Nobel Prize over 100 years ago. I'm also interested in applying these materials, particularly as sensors, and I think that they could have a big impact in fields including healthcare, robotics, and human-computer interaction. I'm looking forward to talking more, and if you have any other ideas for these materials, then I would love to hear them. Thank you.
Hi everybody, I'm James Gabbard, a PhD student with the MIT Van Rees Lab. Flying insects and swimming fish are inspiring a new generation of aerial and underwater robots. Numerical flow simulations are an ideal way to study these propulsion strategies and to test out new biologically inspired designs. But simulating moving surfaces is challenging for CFD packages that rely on a body-fitted mesh. Constant mesh adaptation is expensive, and frequent remeshing reduces the accuracy of simulation results. In my research, I've explored an alternative strategy based on the immersed interface method, which eliminates most meshing costs while maintaining high-order accuracy. Over the past 2 years, I've developed this simulation strategy into a software tool that can tackle a variety of 2D flows with complex moving boundaries. These algorithms will be useful for exploring new design and optimization methods and serve as a jumping-off point for 3D simulations and fluid structure interaction problems. I encourage you to stop by my virtual poster for more details and to check out more flow videos. Thanks for listening.
3D CVIS Kit is a tool we've built that allows scientists to interactively explore 3D oceanographic data in a web browser. Let's take a look at a live demo of this tool in action. We can explore ocean temperature off the coast of Massachusetts using 3D isosurfaces or interactive cross-sections. Moreover, we can visualize velocity data using stream tube plots, such as this one of the Western Mediterranean Sea. Furthermore, 3D CVIS Kit can explore subduction using particle trajectory data. We can also filter trajectories interactively based on the depth that they reached or other criteria. In summary, 3D CVIS Kit is a visualization tool that greatly reduces the mental overhead for exploring 3D oceanographic data and allows scientists to more quickly reach physically meaningful interpretations.